Hello students, welcome back. We're going to continue today with Carbohydrates Part 14. And this will be the last part of Carbohydrates on Test 2. So there's only one more thing I'll need to talk to you about, the carbohydrates here. Okay, and let us begin. Now, normally in the body, you have lots of different types of proteins. Okay, and what I'm going to do here, I'm going to draw. Okay, I'm going to draw this to represent the circulation. And so this will represent circulation. And in circulation, you have lots of different types of proteins. Okay, but eventually those proteins, they will get old. And so when they get too old, you want to destroy those proteins. And so what happens, those proteins will go to the, let's see a good color here. I'm going to make this organ here. See if this makes sense to you. If you can recognize this organ. Anybody have any idea what that might be? That's the liver, exactly. And so what happens, these proteins during normal circulation, they're going to go into, blood is going to go into the liver and go back into major circulation. And when those proteins get into the liver, some of those proteins, let's see here, we're going to have proteins. The old proteins will be destroyed, and the new proteins will continue back into circulation. Okay. Does that part make sense? Okay. You're going in your bloodstream, you're going to have old proteins and you're going to have young proteins. When those proteins go into the liver is going to find the old proteins and destroy them, and it's going to let the new proteins, the young proteins, go back into circulation. Does that make sense so far? Okay. Now, the purpose of this lecture is to explain to you how the liver knows the difference between an old protein and a new protein. Everyone okay with that? Excellent. And so let's do that now. Let's talk about, okay. So I'm going to put here, this is circulation, so you'll know. This is normal circulation. And what is this organ here? The liver. Okay. And now let's draw for ourselves. Let's draw first a very young protein. So here we're going to have, this is going to be a protein. Now when proteins are first made, when they're first made, you're going to glycosylate these proteins. So you're going to put sugars on these proteins. Well, I hope the body's reading that. Right on the end of these sugars, well, that's going to take up too much space. Would you all be upset with me? Instead of drawing the shapes of the sugars, I have a lot of upset people now. Instead of drawing the shapes of the sugars, I'm just going to put here gal for galactose. Okay, you're going to have these galactosyl sugars. Galactose or galactosamine sugars. Now, however, in this state, the liver would destroy this protein. And so what a young protein is going to do is going to protect these sugars so that the liver won't recognize them. And it does this by adding a sialic acid. It's going to add a sialic acid. And that sialic acid residue covers up those galactosyl residues. And now the protein can't be recognized and destroyed. Now, in the liver, let's see what's going on in the liver. So I'm going to put the liver over here. In the liver, there is a protein or receptor that recognizes those galactosyl residues. But now in this state, but if those sialic acids are there, it can't recognize it. So is this protein, once this protein gets to the liver, is the liver going to destroy this protein? No, it's not, because those sialic acid residues are there. Does that make sense? Now what's going to happen? When this protein goes through, as this protein goes through circulation, something is going to happen to it. As it goes through circulation, here we have our protein. 1, 2, 3. I'm just going to put G for our galactose. And then you're going to lose, I'm just going to put S, well, I put SIA for sialic acid still. You're going to lose one of those sialic acid residues. And so now, when it goes to the liver, is this protein going to be destroyed? No, it's not. Because it can't, because those sialic acids, the sialic acids are still protecting or covering up those galactosyl residues. Does the rationale make sense? Okay. Now, as the protein goes through circulation more, okay, you're going to remove more sialic acid residues. Is this protein going to be destroyed by the liver? No, it's not, because it's still protected. And now, as time goes on, it's going to go pass through circulation again, and eventually you're going to have this situation where there are no sialic acid residues covering those galactosyl units. Is this protein going to be destroyed by the liver? Yes, it is. And that's the concept. Could it be simpler? It couldn't be. There's no way to make that simpler. Exactly. So this is how you remove old proteins from circulation, by using carbohydrates. Make sense? Now what we're going to do, we're going to name the receptor. But I want to ask you all something. What does this word mean? I'm just going to write a word here. What is this word mean? Typical. What does that mean? Something that happens normally. Something that happens normally. What if we were to say something were atypical? Not normal. So what does A mean? The opposite of something. The opposite. Very good, very good. So now we're going to name this receptor. Now we could call this receptor we could call it the receptor that binds proteins that don't have sialic acid. Would that be a good name for this protein? No. Why not? It is absolutely too long. Too long. Exactly. Okay? Now, another question for you all. We can give a general name for a protein. What's the general name for a protein that has a sugar attached? What would you call it? So one of you can figure this out, even though I've already done a lecture on it. You can figure, you haven't seen it, but you can figure this out. What would be the name of a protein that has a sugar attached to it? Say that again. A what now? Okay, you said just a general name for a protein that has a sugar attached. A glycoprotein, exactly. And so, can you see that this is a glycoprotein? Glyco means, what does glyco mean? Glycoprotein, exactly. Okay, receptor that binds proteins that don't have sialic acid. Okay? We could also call this the non-sialic acid glycoprotein receptor. The non-sialic acid glycoprotein receptor. Is that a good name? Yeah, it's okay name, but it's still too long. It's still too long. And so scientists have decided, let's just call this, and this is the real one, the A-sialo, because why write non when you can write A? And why write sialic acid when you can just write sialo? The A-sialo glycoprotein receptor. It is. It's still too long. You're right. But that is the name of this receptor. So the acylglycoprotein receptor is responsible for grabbing old proteins because they don't have sialic acid. Grabbing those old glycoproteins that don't have sialic acid, bringing them to liver cells and destroying them. And in that way, we take old proteins out of circulation. Question. What is a glycoprotein? What does the root word glyco mean? Sugar. So a glycoprotein is any protein that has a sugar attached. Any protein that has a sugar attached is a glycoprotein. Question.
Acylglycoprotein receptor is the one that would take in like the last protein?
Exactly. The acylglycoprotein receptor would bind this one. That's what it would bind. Would it bind the first one here? Would it bind this one? Why would it not bind the first one? Exactly, because it has sialic acid on it. And the sialic acid is going to block its attachment to the rest of the glycoprotein. Does that concept make sense? That's the last thing I have to teach you about this test. Unless you all have questions, we're going to wait for questions. Yes?
Is it like a 7-pass receptor?
The acylglycoprotein receptor, is it 7-pass? I don't think it is. I don't remember what it is. I didn't bring my textbook with me. Your textbook has the structure. May I borrow your textbook, please? No, well, I would have to look that up because it doesn't have in your book, in your textbook, it doesn't have what type of protein it is, acylglycoprotein. But I don't think it's 7-pass transmembrane. Yeah, I'll Google it. I'll just Google it at some point. No, I don't want to Google it now. But I'll Google it some other time. Any more questions? Any more questions?
Can you draw mannose? Because I have it drawn two different ways in the notes.
Okay, mannose. The question is, would you draw mannose? D-mannose is I think it's a 2-epimer of glucose. Is that right? It's a 2-epimer. So, if I drew glucose, I'm going to draw glucose first, and then I'm going to erase and draw mannose. CHO, 2, 3, 4, 5, 6. Oh yeah, just CHOH. So glucose would be right, left, right, right. That would be D-glucose. To draw D-mannose, all you do is a 2-epimer is just put your OH over here, and that's mannose. That's D-mannose.
This slide we had it going different, and then on part 7 we had it that way.
Oh, I may have made a mistake. Okay.
This is what we had on part 4.
may have made a mistake on that then.
So it's left.
Right. It's a 2 epigram. Mannose is a 2 epigram glucose. This is mannose. I'm going to put here 2 epigram.
So you said 2 epigram. That means carbon number 2.
Right. It's a 2 epigram of glucose. And so carbon 2 is different than glucose. Carbon 2.
So it's like a 3 epigram. You change.
Carbon 3, exactly. And so what I try to do, I try to always remember glucose, and then I can just draw everything based on it. I remember mannose is a 2 epigram, galactose is a 4 epigram. That's why on the test I say you have to know, they're like those 4 sugars you have to know. And if anything else I want you to draw, I'll tell you it's an epigram of that one. You need to know glucose, you need to know galactose, you need to know fructose, and you need to know ribose. That's another one, you need to know ribose. The 4 that you'll need to know for the test, just know their structure. Glucose, galactose, fructose, and ribose.
And you said, like, if there's a question, you would, if it wasn't one of those 4, you would say, like, draw the 3 epigram.
Right, exactly.
Okay.
For those.
So we don't have to remember that mannose is the 2 epigram of glucose. You're going to tell us draw the 2.
Yeah, for mannose, I wouldn't want you to remember the one that I would want you to remember for epimer would be galactose. I want you to know that galactose. Know that one. But mannose, you won't have to know mannose.
Okay.
You have to remember that. I don't want you to get bogged down with memorizing structures. I want you to understand concepts. Question?
If you want to draw something like the ring form, we gotta say like pureness or sourness.
Only if I asked you to give me the whole name would you have to. If I gave you a structure, and I would say, give me the complete name of this, then you would say pureness of this ring form. But if I want you to draw the ring form of something, then I would put into question, glucopyridose.
How do we know if it's D or L in the ring form? I know beta and alpha.
Now, are you talking about for?
Glucose, D-glucose,
Okay. Now, in the Hayward, let's do Hayward first. If something is in the ring form, how do you know if it's D or? It's the 5 carbon. So all you have to remember, this one is a pretty simple. Yeah, everything is D, but that's what our question was. How do you know if it's D, if it's in the ring form? Yeah. And so, it's the 5th carbon here, so if this is up, if that's up, it's D. And all of ours is going to have, that's going to be up on everything. That's in the Hayward projection. And then, but that's not the hard part. Okay, and then if it's down here, if it's down, that's alpha. If it's up, that's beta. Now, one question that's going to be on the test is going to be this. What if you draw the ring form? What if you draw the ring form of something in the Fisher projection? That's what you should be asking about. How do you know if it's alpha or beta if you're drawing the Fisher projection? It's carbon 1. It's still carbon 1 here.
If it goes right.
See?
Right
in
is
the
the
same
D
direction
or
as
is
the
it
line.
alpha?
If it's in the same direction here, if these two are in the same direction, what did I say in the notes? I think that's alpha. And if it goes the other way, that's beta. That's Fisher projection. And that's carbon number, of course, that's carbon number 1 because that's the anomeric carbon. So if it's on the same. Because 5 is going up. 5 is going up, that's D. And see, if we had some actual models and you made yourself a D sugar, and that 5th carbon is going over this way, when you fold that around to make a ring, that OH is going to the right, when it flips up, it's going to end up going up. I guess I should have, when we were going over in lab, if you would have noticed that, it's going to make itself go up when it flips around. So that makes it D. Okay. Very good. Okay, study session tomorrow at what time? 10. Very good. Have a great day, you all. Have a great day. Don't unplug the...
I sent on the